Welcome to today's lecture on advanced techniques in microbiology. In this session, we will delve into a wide range of topics that are crucial for understanding and applying cutting-edge methods in the field. These techniques have revolutionized the way we diagnose infections, investigate outbreaks, identify microbial species, study host pathogen interactions, and much more. So, let's dive right in and explore the exciting world of advanced microbiology techniques. Real-time PCR is a molecular technique that quantifies pathogenic microorganisms in clinical samples. It monitors amplification in real time using a fluorescent reporter system. It provides precise and early diagnosis of infections with high sensitivity, specificity, and rapid turnaround time. The use of whole genome sequencing, WGS, in outbreak investigations enables us to analyze the genetic makeup of pathogens, aiding in tracing the source and transmission routes of infections. This advanced technique revolutionizes epidemiology and enhances public health efforts. Mass spectrometry-based proteomics has emerged as a rapid and high-throughput method for identifying microbial species. By analyzing the protein profiles of microorganisms, this technique allows for quick and accurate identification, often within seconds. Matrix-assisted laser desorption/ionization time of flight, MALDI-TOF, mass spectrometry is particularly popular in clinical microbiology laboratories. MALDI-TOF can distinguish between bacterial and fungal pathogens, aiding in the selection of appropriate antimicrobial therapy. This technique has significantly reduced the time required for microbial identification, enabling timely and targeted treatment. Flow cytometry is an advanced technique that characterizes and quantifies immune responses to infectious agents at the single-cell level. By employing fluorescently labeled antibodies, flow cytometry enables the identification and characterization of different immune cell subsets involved in host pathogen interactions. This technique provides valuable insights into the immune system's response to infections, helping researchers understand disease mechanisms and identify potential therapeutic targets. Flow cytometry also allows for the analysis of multiple parameters simultaneously, enhancing the depth of immunophenotyping studies. Continuing from our discussion on mass spectrometry-based proteomics, we focus specifically on matrix-assisted laser desorption/ionization time of flight, MALDI-TOF, mass spectrometry and the rapid identification of bacterial and fungal pathogens. MALDI-TOF is a powerful tool that relies on the unique mass-to-charge ratios of microbial proteins, enabling the identification of microorganisms from cultured isolates. By comparing their protein profiles to extensive reference databases, MALDI-TOF can provide reliable and timely identification results, allowing for appropriate and targeted treatment decisions. To understand viral infections, researchers culture and isolate viral pathogens from clinical samples. This involves techniques like cell culture systems using susceptible cell lines. Researchers create optimal conditions to study viral replication, morphology, replication dynamics, and interactions with the host. Virus isolation and propagation aid in diagnostics, drug development, and vaccine creation. Bloodstream infections pose a significant threat to patients, requiring rapid and accurate detection for effective treatment. Automated blood culture systems have revolutionized the microbiological diagnosis of bloodstream infections. These systems utilize specialized blood culture bottles containing growth media that can support the growth of microorganisms. They also incorporate automated instruments that monitor culture bottles for microbial growth using various detection techniques. The advanced systems can provide early detection, allowing for prompt intervention and targeted antimicrobial therapy. In this advanced topic, we will explore genotypic methods, including DNA sequencing and PCR, used to identify and characterize antibiotic resistance genes and mechanisms in microbial pathogens. These techniques are crucial for monitoring and combating the rise of drug-resistant pathogens. DNA sequencing allows the identification of specific genetic mutations or acquired resistance genes associated with resistance, while PCR assays enable rapid and targeted detection of resistance genes. By using these genotypic methods, researchers can develop effective strategies to combat antibiotic resistance effectively. Advancements in molecular techniques, like multiplex PCR and microarray analysis, have improved the detection and characterization of antibiotic resistance genes. These methods allow for simultaneous detection of multiple genes, aiding in tailored treatment strategies and infection control. In the video, we will explore emerging molecular techniques, such as next-generation sequencing and metagenomics, which have revolutionized viral diagnostics. These methods allow for the rapid detection and identification of viral pathogens with unprecedented sensitivity and resolution. Microbial biofilms are complex communities of microorganisms attached to surfaces, known for their resilience and resistance to antimicrobial agents. Studying biofilms requires advanced microscopy, molecular, and genetic techniques. Confocal laser scanning microscopy allows visualization of biofilm architecture and microorganism dynamics. Molecular techniques like transcriptomics and proteomics unravel genetic makeup and behaviors of biofilm-forming organisms. These approaches shed light on biofilm formation, antimicrobial resistance mechanisms, and strategies for controlling biofilm-related infections. High-throughput screening methods, like high-content imaging and robotic assays, are valuable tools for discovering antiviral drugs. They allow for rapid analysis of compounds' effects on viral replication and cell viability, helping to identify potential treatments faster. Advanced microbiology techniques, such as real-time PCR and whole genome sequencing, play a vital role in diagnosing infectious diseases, studying outbreaks, analyzing pathogens, understanding host interactions, and developing new treatments. As future healthcare providers, your proficiency in these techniques is crucial for improving patient care.